1 minute more. Wait for it. So the first speaker for this session is from Saidullah Kushara, the backbone behind the organizing committee of this event for which we are here. It's a great pleasure to introduce a very great scientist and a real colleague of mine. He has over 200 publications with impact factor of over 700. And he's a well-known scientist in the area of bioanalytical mass spectrometry. He is recipient of several national and international awards, including the Civil Award of Pakistan. And he is a member of Global Young Academy and the selected member of class. And he has done various contributions and a very well-known top scientist in the field of mass spectrometry in the globe. So let's welcome, with a warm welcome to Dr. Mushar.
Thank you very much, Vanita, for your kind words. So, good afternoon to everyone. The topic which I'm going to talk about is very much related to the application of mass spectrometry to deal with the natural product samples, the natural product, plant material, and the samples, as they are quite complex. So that's what I'm going to talk about in my lecture. I think since last 2 days, we are listening about the importance of natural products, and especially the compounds that are isolated from the natural product. So you can see in this slide, since 1981 to 2014, several drugs and lead molecules have been isolated from plants. Some of them are using as it is for the treatment, and some of they are the natural mimics, and some of the derivatives you can see. So this actually slides highlights the importance of natural product and why it's important to still continue the research or explore the new lead molecule from plants or from the natural product. But when you are going to deal with the plant and the natural product, there are several challenges. So if you just look this first part, that redundant isolation of previously known compound. This is one of the big issue when you are going to work on the plant side. So you can just look these two molecules as an example. These are very recent data that I have taken yesterday from the Science Finder. The beta-cetosterol, was so far 4,233 times isolated and characterized. This is something it's not a good news, but unfortunately, the unavailability of the methods, the complexity of natural product, the people are re-isolating again and again a molecule which is very well known, and they used to characterize and spend time and money. Just a similar example in gallic acid you can see. So this is one of the important issue as far as the natural product chemistry concerned, particularly from the plant side. And someone is reported that it actually costs around 50,000 to identify a one lead molecule from a plant source. So you cannot spare a time, especially to re-isolate and characterize again and again. Another important issue, the massive utilization of raw material. If you are going to characterize a compound from plant side, it needs to be, in tons of tons, unfortunately, because the natural abundance of the compound is quite low. So that's actually utilize a massive plant material. So how we can restrict, the use of such unnecessary utilization of plant material. As far as the product of plant material concerned, we all are familiar that they are herbal products. So herbal products we all are used to use, but quite complex as far as their standardization concerned. So the analytical methods are still required to handle such type of complex product. So that's what I'm going to talk about, that how we can see a technique, particularly mass spectrometric technique, to resolve all these issues. That's what I'm going to talk about. Very shortly, why mass spectrometry? The beauty of mass spectrometry is, it's a buffet of several ionization techniques. You can deal from very non-polar compound to the highly water-soluble compounds. The diversity of ionization techniques is outstanding. It's a true hyphenated technique connected with GC, LC, or CE. You can just name. So the mass spec is there. Another important thing that the hybrid system, I mean the coupling of different mass analyzers, which actually provides a high sensitivity and resolution. Another important thing is now the imaging technique. The people are using just a mass tool for the detection of different disease biomarker during the operation and all this thing. So very brief justification that why this mass spectrometry is nowadays getting familiar day by day to resolve different issues. So in my presentation, I'm going to talk in three aspects of application of mass spectrometry, that how it can face the challenge related to phytochemical complexation. The first part where I'm going to deal the high throughput method development that we have recently developed and applied to various plants for different purposes. The second part is related to the standardization of polyherbal formulation, that how the mass spectrometric tools can be useful to handle such type of complex. The third part, if the time allows, I will talk about that. Nowadays, the people are moving towards the bioassay screening based on mass spectrometry. Because of its high sensitivity, it's limited the use of very low concentration of substrate and enzyme. So these three applications I'm going to cover in my upcoming slides. So this was one of the methods that we have developed. Is based on laser desorption ionization. It's actually, if you just add the matrix, it's a matrix assisted laser desorption. But in this method, we have not used the method. And it's a very simple and high throughput method. Believe me, just requires a very few milligram of plant material. Just need to make it powder and sieve it. The finest part of the particle placed with the help of solvent, a non-polar solvent like dichloromethane or something, and just applied laser. So we have used the NdYAG lasers, and with the help of this, we have identified so many compounds in a very high throughput and a very quick way for the identification of compounds. So you can see that these actually the details can be seen from the paper, but you can see that we have applied a different solvents, and actually solvents destroy the outer layer of the plant material. And that's allowed to ionize the compound from the the inner side of the plant. And we have screened this application with leaves, roots, and seeds. Unfortunately, the developed method is not very well valid with the seeds, because so obvious the outer part of the seed is quite hard. So the method is not that good enough. But as far as the leaves concerned, they are good enough to detect the ions. Similarly, you can see the picture, and you can see the 550 micron particle and 100 micron particles were good enough to be analyzed with the help of laser desorption ionization. So now, after developing a method with so many things that you can see, we have screened a large number of plant material. Mostly these were leaves, and you can see that the ions difference was so significant. Which supposed to be, because if we are talking about the secondary metabolite, it varies from plant to plant. So the fingerprinting was very much promising and very much characteristic with respect to the already known metabolites. I just want to show the example of the analysis of Nicotiana leaves. We know that these leaves are known for the nicotine, and now you can see this is the leaf laser desorption ionization fingerprint, and in that leaf we have identified the nicotine. This is the standard lift experiment, MSM experiment, and that is the done which we have recorded in the leaf part. So this is believe me, it was just took 5 minutes to record and to analyze. So this is the high throughput method that we have developed. No extraction, nothing at all, just a few microgram of plant material subjected to laser desorption ionization and that analysis as well. So very high throughput way, and I'm pleased to share that after publishing this part, the people have using the same method. Like for example, in case of this paper, the people have used the lichen, the same that the method that we have developed and published this in a good journal, analytical one. So that method was related to the laser desorption, a high throughput way that how you identify. So the second part, actually, we have focused more on the liquid chromatography ESI MSMS method development. And that is the method where we have named as a two-stage protocol. In case of plant, the first objective to develop a chemical fingerprint, obviously to identify the number of metabolites associated with particular plant species. And the second objective is to quantify it. So with the help of stage 2 protocol, you can not only develop a chemical fingerprint, but you can also use the similar fingerprint for the quantification of the compound which you want to quantify. Either it could be biomarker or could not be a biomarker, just a marker compound. So the stage 2 protocol that we have developed, but the beauty of this stage 2 protocol is about the very accurate identification of compounds. Particularly, it's a drawback of mass spectrometry if it is going to be handled by the non-mass spec person. It may identify false positive compounds. So it has to be go through with very stricted and stringent criteria if you are going to declare the compound is that, because you don't have anything physical in your hand. So you are just applying the mass spec tool for their identification. So in that case, we have applied a very high level of confidence tools for the very accurate identification. For example, the first stage was high resolution data. We also developed a reported literature and the one Excel sheet that reported compounds, particularly from the plant species. Then we have focused and recorded the tender mass spectrometry of each analyte, and then try to identify the matching if there is any mass database available. This is the fourth stage. The last stage, which actually gives you the stage 1 level identification, is the use of the standard, which is very obvious that any other technique you have a standard that is so obvious to try to identify. So we have used very strict criteria for the identification, including the match with the ppm error and the isotopic pattern and the MSMS pattern. So these actually criteria that we have developed and published, and believe me, this methodology was very useful, and currently we are using a similar methodology as far as the analysis of the plant extracts and things are concerned. So by applying the same developed method that we have discussed previously, we analyze different types of samples. For example, you can see that we have analyzed a large number of metabolites in salvia species. So around 5 salvia species, we all know that the salvia species are very well known as far as their biological value concerned. So we have analyzed the different salvia species which are actually very close in terms of morphology. So these two slides actually showing the difference of metabolites associated with each species. You can see that this is a positive mode ionization method, and these two compounds, particularly very specific to this species, similarly just move on. So this is positive based metabolite identification protocol, and similarly this is negative based identification protocol. So it's up to you whether you want to see the similarity between the species or differences. Although we have analyzed both, but just mention these two slides. Another paper that you can see is related to the cucurbitaceae families, a different species of these genera. We have around over dozens of species actually we have analyzed with the help of developed method, and again identified a large number of metabolites including different cucurbitaceae in the same species, in various species. And what I would like to say here, particularly, if you just look this part, it's mentioned as a pool. The pool, what's the meaning of pool and importance of pool? For example, if you're going to analyze a 5 species and just make a one pool, believe me, it provides a kind of quality control, because the herbal products doesn't have a very authentic standard. So for quality control, especially in the crude, I would recommend to use a pool sample of the sample that you are using. It gives you a kind of confirmation of each analysis. So we have successfully utilized a pool sample, especially for the quality control, identify large number of compounds, and some of them we quantified. So now proceed to the qualification data. Another important application that we have used by the developed method about the how to identify the contamination, cross-contamination of plant material. You know, it's quite common and it's a big issue as far as the herbal raw material concerned. Contamination of kind of another herbal material is fine. The contamination of toxic plant to the herbal material is quite risky. You know, the method is really much needed, to identify the cross contamination, even the minor contamination. So this is the contamination. Actually, the one of the Session species was very toxic and have very similar kind of morphological appearance with the herbal plant, which is Zizubus jujuba. So actually, we have selected this Session species, a different Session species, which is available in Pakistan, and try to identify. So this is the metabolic level separation with the help of these scatter plots. So with the identification of compounds, all three species were nicely separated. And after that, we have tried to intentionally mix the different ratios of toxic species with the medicinal plants, and able to identify even less than 1% of the contamination of toxic plant into the medicinal plant based on the biomarker peak that we have identified in the plant material. Okay, so this is one of the application where you can use a high throughput method for the cross contamination identification of toxic or any other. So these are some publications related to the method that we have developed. Third part is again, just keep remind my topic. The topic is related to how to handle the complexity of plant material. So this is very new, application that we have explored. Is the development of lipid chromatography, high resolution ESI-MS database development of natural product. In mass spectrometry, if you are the user of mass spec, you know that the compound can be identified with the help of database. But that database is very much restricted with the EI. And EI is only compatible with the GC. And GC is subjected to the volatile compound. So if you have a kind of polar compound, so the database application is very much limited. And very few databases are actually available by which you can compare. So this is the initiative that we have started at least 5 years before to develop a database of natural products. But the question is that why we need to do and why we are unique in that. This answer goes to this molecular bank. Fortunately, our center is having a kind of molecular bank, and molecular bank is actually a repository of thousands of purified and characterized compounds. In addition to that, we have around thousands of compounds actually from the natural product side. So that was very unique natural products, not commercially available. So that was the basic reason that we have started to work on the development of electroionization database library for our own purpose or for commercial application, right? So this is the thing that, and you can, if you just look the entry of our molecular bank is quite comprehensive. We have all information from where it's isolated, what its mass, what's the purity, so everything is there. So so development of such type of database, especially in case of thousands of database, you need to have a smart strategy. You cannot run the thousands of compounds alone. So what we have done, we just take a group photo. This is like a one photo accompanies so many people. So then we have developed a pooling strategy, a very small strategy, a single run HPLC strategy by which we have analyzed over hundreds of compounds in a single run. So the pooling strategy was very smart that we have developed. The compounds actually were pooled together based on two important things. The one is pK value, the another one the masses. We just try to avoid to pool same masses in a one pool. Okay, so that was the strategy that we have used for developing the pools of natural product. And with the help of this, we have recorded the spectra at various collision energy. You can see average collision energy. In addition to that, 10, 20, 30, 40 In order to make it reproducible, in order to make it more widely applicable to a variety of data obtained from various instruments. So this is the paper I think just published this year or maybe last year, and you can see that it's a pool of natural products, particularly alkaloids, and they were nicely separated with a very sharp piece. So that is the application, that how you can even standard pool quickly develop an LC profile for their rapid identification in plants. So remember that I have talked about the development of database. So if you just look example, so we have recorded the spectra, just a spectra of different compounds, and then develop a window compatible profile tool. This database is really good for searching of the compound, because the all natural product mass spectra is built in that database. Okay, so this is actually done by our students. So the database, we just look, we have not commercialized that, yes, but you can look that you can enter as a the student or guest. And this is the screen by which you need to add the details of your experiment, the preparation and whatever. So after entering this and putting the spectra, you can identify the compound with very high confidence. You can see that this is one of the alkaloid is actually from our database, and you can see that the spectra at various collision energy. So it's no matter at what collision energy you are going to record, the spectra is there. The spectra is already built in our database. So this is something very strong feature which actually make more commercialized and more saleable in future, inshallah, this database. So after developing database, we have applied database to the analysis of various class of compounds. This is one of the examples where you can look that we have analyzed a broad range of alkaloids, and over 200 alkaloids database that we have generated with the same methodology, and screen the same method to a variety of plant extract and identify the compound with the same pattern. You can see this is the sample in the plant extract, and this is the reference database, how closely and exactly they are matched. So this is the way that you can accurately identify compounds without any false positive in a very complex sample like plant extract. And I'm pleased to share that this work was published and considered as a cover photo of one of the prestigious journal last year. So this is very nice art made by one of my students. Anyways, so remember the database project is not too old in my lab. We just started 5 years back and recently ended up with some excellent finding. Now we are applying this database to different classes of natural products for their rapid profiling and for their rapid identification. This is one of the applications. I have only, I think, 8 minutes. So very quickly, the second part, the most important is the polyherbal standardization. Remember, I told you that how we can standardize a polyherbal extract with the help of mass spec. With this, actually, we worked with the Herbion company. The latest is one of the very selling item of Herbion, useful for the cough and asthma purpose. So this is a mixture of over 10 herbs. So they were very much keen to standardize this product and to add some value addition for their cost. So we have worked with this. I just want to show the example of one raw material, but this strategy is very important that we have published, identified and published in Metabolomics. So what we have done the first step, the collection of plant raw material by ourselves. We have not relied on the company, actually. We have just collected the plant material from various locations, Pakistan. So I think over 10 sites that we have collected a raw material, then try to develop their LCSI profile or fingerprint to see whether they are reproducible or not. The statistical analysis, then we have focused on the biomarker component, and then we have tried to purify those biomarker components, followed by the quantification. So this is the strategy along with the objectives of each step, and finally you may lead to the herbal standardization phase. So now, as I mentioned, that the one plant I would like to discuss here, this is Adota vesica. It's a very well-known plant, widely distributed in Indo-Pak region. So remember that we, as I mentioned, the first step was to collect the raw material from different parts of the country, starting from Punjab, the Balochistan, and then Kashmir side, and try to develop the fingerprint. This is the LCSI MSMS profile of all these 14 accessions. And you can see that they are quite reproducible, apparently. The peaks, the LC profiles were very close, which is a good sign, which means that the plant material itself doesn't have a kind of influence from the environment side or some other factors. But they do have, but generally they were very close. And now you can see that we have applied a very extensive statistical analysis to see the distribution of metabolites within the 14 samples. And you can see that the pictures were really cool. I mean, like they were somehow different, but not that much. The all 14 species were somehow having a similar metabolite, except the difference of their concentration of few metabolites. So these are the different statistical. Actually, we have applied a very stringent and strict statistical tools to see the different views of the things. Without going to detail, so as I have mentioned, the objective was to identify and standardize the thing. Now you can see this is the lincus, which is actually the extract of 10 material, and this is the Abrota vesica. And we have identified these 3 biomarker components rightly, starting from Abrota vesica till the final product. And these were known compounds, but the method was not developed to see them. So with the help of this method, we have identified these 3. And now you can see this is the extensive LCSI MSMS profile. And these were the 4 compounds which we have actually identified and purified and quantified in all that plant extracts and the herbal products. Yes. So now you can see after the quantification of these 4 biomarker components in all plant material that we have collected, we have noticed that in Pakistan, Pakistani raw material, especially collected from these side in the Kashmir, will have the high concentration of viscosine and viscisinone. As far as the India has reported the highest concentration at this site, which means that the plant raw material is indeed a double fold of the already known one, the good quality to consider or to prepare the herbal extract. And we have also quantified and validate these things in the samples as well. Okay, in just 2 minutes, I would like to share you that how you can use the mass spec for the bioassay screening purpose, the rapid method. We have selected the particular activities related to angiotensin converting enzyme. The activity is responsible for the blood pressure and all these things. So we have monitored the conversion of angiotensin 1 into angiotensin 2 with the help of quantification using triple quadruple mass spectrometry. So that is the method that we have developed, and with the help of the method, we can also calculate the IC50 of the compound. So if you just look at these 2 standards that we have screened, the captopril and lisinopril, and an excellent regression curve that we have developed and identified the limit of detection and limit of quantification as well. But most importantly, if you just look the comparison, this is our developed method, and this is the method reported so far, analytical method. I'm talking about the spectrophotometric method. So the method that we have developed was outstanding in terms of sensitivity, in terms of the usage of enzymes amount, substrate. So that is the way why the people are moving toward the mass spec for even for the screening or handling the complexity of natural products associated with enzyme screening. So with this, I'm very thankful to various funding agencies who are involved with us, OPCW, Higher Education Commission, the ICT R&D. This is actually currently known as Ignite. And these are the students. Dr. Aslan is one of my former students currently working here as an assistant professor. Dr. Amna Jabbar is currently she's research officer. These are my former PhD students, Saeed and Adipa, Zarina. These are Zaki, and these all students are currently working in my research group. So with this, thank you.
Time for 1 or 2 quick questions. very quickly.
Thank you very much for this important lecture. I'd like to ask two quick questions, actually. For the database, are you going to add the other spectroscopic data like NMR, for example, for your compounds? And the other question of the
Okay. Yes, it's our future plan. Obviously, when you are going to record mass spec data, it's quite complementary. It would be, good to have NMR data as well. The fortunate thing is that the database, as far as the NMR data is already recorded, we just need to integrate. So, because, as I've mentioned that if you're going to talk about the molecular bank, the compound is already characterized, very well recorded. So the NMR data is already there. We just need to integrate, but in future, yes.
And the other quick question also was like, does the database will going to work with other ionization techniques?
No, it has to be ESI because the compounds which we have analyzed is quite polar enough to handle by ESI. So it is only compatible with the ESI. I mean, you cannot see because the fragmentation pattern induced from EI is bit different in comparison with ESI. Sometimes, sometimes they may have a common fragment, but these two databases are not matchable. And there is a lacking, particularly in ESSI. That's why my research group is contributing in that part.
Just one comment. I think this work of yours is very much useful for the industry, like industrial application, like adulteration of food, etc. So, like, what are the steps you have taken to involve them more? At least for them to know that you are working on this data.
Yeah, this is a very, long brief question is, to be answered. The problem, the industry, especially in Pakistan, they're still not very well familiar the beauty of the high-tech instruments. They are not very well, ready to adopt the new technology unless they have to, they need to be pressurized. So that is the issue. But anyway, as I mentioned that the Herbion is already, now they are familiar the importance of the findings that we have. Similarly, Hamdard is also working with me. The one of my projects is actually not discussed, but we are trying to develop a chemical fingerprint of plant extract, just a plant extract. You can identify the plant with the help of extract. So we are developing a plant extract database, not the natural product, but the plant extract database like some impression of each, individual. Yeah.
Thank you very much. Speaker, what the session is, Professor Sami Abazif, the professor and head of pharmacology department at the Faculty of Pharmacy, University, Egypt. He obtained his PhD in pharmaceutical science from Faculty of Pharmacy, Cairo. He obtained his MSc and PhD from the Graduate School of Pharmaceutical Sciences from University of Tokyo. He has done his postdoctoral degree at HED Research Institute under the supervision of Professor Iqbal Chaudhary. He has supervised about 25 MPhil and PhD students, and he's going to talk about on chemical and biological studies on ligands, and we are ready to hear you, Dr. Sami.
Thank you, Chair, very much. It's my pleasure to be here. Actually, it's difficult to speak up after Musharraf, my friend Musharraf, but I will try to beat him in Karachi. First of all, I'd like to really express my thanks and gratitude to Professor Atar Rahman, to Dr. Iqbal, whom I am proud to be one of the alumni of his group in 2008. to all of the brothers and sisters in ICCPS, a great respect and appreciation from Egypt to all of you. Today I'm going to speak about flavonolignanes, some of the chemical and biological studies which I have done in Egypt about flavonolignanes. I will focus on the model species producing this class of compounds, which has been studied for hundreds of years and which has been used for treatment of liver diseases for hundreds of years, milk thistle or Silphum marianum, which is an annual or biennial plant in its life cycle, native to the Mediterranean region. And the silmarine is, the word silmarine refers to the standardized fruit extract, which is available in many pharmaceutical products around the world for treatment of liver diseases, and recently as a chemopreventive agent for prostate carcinoma. To understand the economical importance of this plant or the product taken from this plant, we can see here the upper graph. These columns represent the sales of dietary supplements made from medicinal plants in the United States from the year 2000 until 2020. As you can see, there is an increase over the years, which reaches now almost 11.6 billion US dollars. Silphum marianum and its products represent the 23rd position among these dietary supplement products, with a sales figure more than 19.8 million US dollars, with 8 in 2020, with 8.4% increase from the previous year. The chemical description of flavonolignanes, this class of compounds, today's topic, from this plant has begun actually early in the middle of the 20th century, when Chandler detected flavonoids in the tincture made from the fruit of this plant. Years later, Wagner isolated pharmacologically active fraction from this plant. A few years later, actually, Pelter and Hansel isolated a compound called silibene and gave its empirical formula. 70s and 80s, the isomers of silibenes, like isosilcristine and isosilcristine, silidionine, and isosilbenes were described. And later, in 2003, Lee and Leo from North Carolina University in the United States describes the molecular structure and stereochemistry of these compounds. In the next slide, you can see the main flavonolignanes produced by the major variety of Silphum marianum or milk thistle. As you can see the structure here, some of them are diastereomeric isomers, some of them are regioisomeric forms, others are structural isomers, and taxophylline is the major or the main flavonoid precursor from which all of these flavonolignanes are formed. All of these flavonolignanes, all of these compounds are attributed to the purple flower variety marianum, which is a major variety used in the world nowadays. Another variety which is available in Egypt and also other parts of the world, variety albiflora, produce similar types of flavonolignanes based on the flavonone eriodactyol, namely isocyanidrine, silyhermine, silandrines, and silimonene. This is called the variety albiflora, and it is characterized by the white flowered plant. We began the work actually by extraction of silimarine, or these components, these flavonolignanes contained in silimarine, constituting the major fraction of silimarine from the fruits of this plant, by separating the pericarp from the kernel of the seed. Pericarp, which is the cover of the fruit, from the kernel of the seed, by a locally patented method to get a more hydrophilic, a more soluble, and an extract of silmarine containing more flavonolignanes. While from the kernel, we can produce the pure oil of silver. Then we began extraction of silmarine from the pericarp of the fruits by using a method called ASE, or accelerated solvent extraction, to the contrary of the conventional method, which is successive extraction, which is based on two-step production. First, defatting, removing the fats from the seeds by treatment with normal hexane for 8 hours. Then extraction of silmarine by methanol for 5 hours. Takes a long time, generates a lot of amount of solvent, or uses a lot of amount of solvent, generates waste, and silmarine can be lost in the defatting step, while our method is characterized by reduced time, reduced amount of solvent, extracting the material under pressure, increasing the dynamic side of extraction, the contact between the solvent and the plant material. Then we try to study by response surface methodology the main factors affecting the extraction process. These were the time and temperature, and we get a hot spot here, the best condition to get silmarine, the optimum condition, the optimum temperature, and optimum time to get highest amount of silmarine, using these times, 30 minutes, 60 minutes, 90 minutes, and using different temperature ranges. In the responses, we use two types of response to measure flavonolignanes. We use the UV absorption and quantitative NMR method, will be elaborated later in this presentation. Then we get a very similar results from both methods, from QNMR and UV methods. Our method, or our proposed method for extraction, optimized method, actually is characterized by not using the petroleum ether or hexane, the very non-polar solvent for use for defatting, and using much less time in the extraction step with methanol compared to the conventional method, which is prescribed in the European pharmacology. Then we try to make a standardization for our extract to know how much flavonolignanes are contained in this extract by using two orthogonal method of analysis. First, we developed an HPLC method, a validated method for, as you can see here, a baseline separation of all of the components, very similar components of silmarine, silibine A, silibine B, isoleucine A, isoleucine B, silibionine, only compound which you have some overlap with other small peaks, silicrystine A, isoleucine, and the major or the precursor flavonoid taxophylline. We also used quantitative NMR method as orthogonal to HPLC, depending on the authentic silibine with the mixture of silibine A and silibine B. We developed NMR spectrum from increasing concentration of silibine, and we quantified the integral value for this proton attached to carbon 2 in the flavonoid part of the flavonolignanes to get our standard curve and compare it with the HPLC method. Then we use this NMR method actually to compare the amount of silmarine, amount of flavonolignanes obtained by extraction, either the whole fruits or the pericarp. You can find here NMR spectra for the extract obtained from the whole fruits, and the one behind the extract obtained from the pericarp. We have used quantitative NMR parameters, the single 90 degrees pulse experiment, which offers magnetization for all of the nuclei we have, and increase the relaxation delay to 60 seconds to account for the relaxation of all nuclei we have for the compounds in the extracts. And then we can obtain a good result. Actually, almost double amount was produced from flavonolignanes or silmarine in the extract produced from the pericarp. We have used also the same HPLC results and NMR quantitative NMR methods to quantify the silmarine contained in different sites in Egypt. From the south to the north, we obtained here characterized the different varieties available in this place, characterize the color of the fruits and other morphological characters, and most importantly, the content of silimarine in different populations from which Silybum marianum can be collected all over the country. By using HPLC and NMR, we can obtain the content of the individual components present in the different population. Then we apply chemometric analysis, principal component analysis, and agglomerative hierarchical clustering to obtain mainly 3 clusters here from the principal component analysis, also confirmed from the agglomerative hierarchical clustering from our populations of the plant growing in Egypt. Then we can obtain these. Then we can generate these hypotheses out of the chemometric analysis. We can have 3 populations clustered into 3 classes. Silimarine rich population, population with low silimarine content, less than 18.8 milligram, and populations with higher silimarine content, higher than 18.8 milligram. Generally speaking, the population of Silybum marianum growing in the Nile Delta were highest in silimarine. This was known, or this was expected, as the plant is native to the Mediterranean and the delta is near the Mediterranean area. No correlation was found between the fruit color and the silimarine content, and this was very important because some people were thinking, or they have a common thought, that the darker fruits containing more silimarine. And the most importantly, the last 2 hypotheses here, we found a high correlation between silybin A, silybin B, and silicrystine, and a little bit lower correlation, or some correlation to a lesser extent, as we can see the quantitative data now, between another 3 components of another 3 flavonolignanes inside the silimarine, silidimine, isosilicrystine, and isosilipine. As you can see, we can have now 2 main populations. A population rich in silybin A and silybin B, along with silicrystine. Another population rich in silidimine, which is this peak, and the other components are very low. Silicrystine is low compared to the upper one. Silidimine was also accompanied by isosilicrystine and silybin B. So, upon obtaining this results, we think that we can have 2 chemotypes of Silybum marianum growing all over Egypt, and also we found it growing all over. The same theory or same hypothesis can apply to Silybum marianum population growing anywhere in the world. We think backward. We think about the biosynthesis of this compound inside the plant, and we examine the correlation in a quantitative manner. As you can see here, the pie plot containing showing the correlation between the variables, the 3 vectors of isosilipine B, isosilicrystine, silidimine, very near to each other. The 3 vectors of silybin A, silybin B, and silicrystine were also much more near to each other. Actually, isosilipine A was a little bit controversial because it was near silybin A and B and away from its very related isomer isosilipine. This can be shown on the correlation matrix in a quantitative way. As you can see here, the correlation between silybin A and B almost 1, and also 0.999 correlation between silybin B and silicrystine A. The same correlation, very high correlation between silybin A and silybin. A little bit lower correlation, but still there are positive correlation between isosilicrystine, silidimine, and isosilipine B. From this quantitative data, from this chemometric analysis, we think backward, as I said, about the biosynthesis of this compound. We think that we have to research how these flavonolignanes are produced by the plant, are biosynthesized by the plant, and the common pathway for biosynthesis of these components, flavonolignin, was known for many years ago to occur by coupling, oxidative coupling, actually, between a radical formed from coniferyl alcohol and a resonance stabilized radical formed from taxipholene. And according to which radical here is used, we will have different flavonolignin. So we can imagine our story, it will go like this. In the silibine-rich populations, silibine A and silibine B, this radical, which is formed by radicalization of hydroxyl group on carbon number 4 prime, or each associated radical, will give rise to silibine A and silibine B. While the plant, if it uses the other radicals here, formed from the radicalization or loss of proton and electron from the hydroxyl group on carbon number 3, I'm sorry, it will give rise to the other set of flavonolignin, silidinine, isosiliclystine, and silibine. So we can have a global image about the biosynthesis of these magnificent molecules, biologically active molecules, and we can say, starting from the common pathway for biosynthesis of flavonolignin, we can have either taxipholene or eridactylol. Eridactylol will give rise to the flavonolignin inside the white flower variety, while taxipholene will give rise to flavonolignin, actually two chemotypes from this plant, from the purple flower plant variety Marianum, depending on which radical will be used. Actually, the stability of this radical the question here actually is okay. Then it was very interesting for us to go for the literature and see what other flavonolignin are produced by Silva marianum or other kinds of plants which can be integrated inside our hypothesis. We found a very nice molecule called seli-amandine. It was only published in one publication by a group from New Zealand in 2007 in Planta Medica, and it was supposed to be this compound, seli-amandine, a decomposition product from the well-known silidinine by aging of the extract or aging of the tincture of Silva marianum. Unfortunately, when we look back for the literature, which was a letter in Planta Medica in 2007, we found a little bit discrepancy in the isolation or the analysis, HPLC analysis of this compound, and we tried to conflict or try to confront this pathway, confront the idea that seli-amandine is not a natural compound, but it is a decomposition product. We went back to search for our NMR data, quantitative NMR data. We stacked the spectra together, and we looked for the peaks of seli-amandine. Of course, we first isolated seli-amandine and then got its pure spectra. And then we found a very good correlation between this compound and both silidinine and isosiliclystine. And then we tried to make hypothesis for the biosynthesis of seli-amandine in the same way as other flavonolignin are formed, specifically isosiliclystine by ketoenol tautomerase, followed by peroxidation, followed by lactone formation to give seli-amandine, and we propose here seli-amandine as a natural component inside silimarine, not a decomposition product as this paper was saying. Then finally, I will elaborate on the two varieties of Silva marianum growing worldwide. Actually, the major one, which is the purple flower, and The other one, which is a bit more rare than the major one, variety albiflora. We make, as you can see here, the dashed chromatogram in front, showing the kinds of flavonoid glands produced by variety albiflorum is quite different from that produced by the variety Mariana. We try to characterize this by TLC. The first lane showing here extract from the fruits of variety of albiflora, and then lane 2 and 3 showing the isolated compounds, isocilandrine and silihermine, while in lane 4 can show the extract from the fruits of the common or the major variety Mariana, with its component here also isolated. So we characterize this by TLC. We try to make authentication by a rapid analysis to make genetic authentication. We found a little bit difference in the proximity matrix here in number 1, lane 1 here, by using different primers, short primers. We found the white flowered variety is clustered a little bit away from the 2 and 3, which are the purple flowered variety. We try to make finally biological characterization also of this white flower variety, which is with different kinds of flavonoid glands produced. So we went to some cytotoxicity and safety experiments, but not shown here. And then we did in vivo experiment. We chose the actual hepatoprotective property of this extract produced from this plant, because it's the very well-known use and the very well-known activity for silimarine. We did the experiment on rats with paracetamol using a high dose to induce the liver damage, and then we measured the liver function biomarkers and oxidative stress biomarkers, NADA or glutathione or superoxide dismutase, and also made some histopathological examination for liver sections. You can find here the white flower extract almost was the same as normal. They don't produce any kind of harm to the liver cells, while paracetamol here, the black one, increased the enzymatic functions of the liver, this bilirubin also. And then after that, when the white flower extract was used in different doses, it produced dose-dependent decrease in the oxidative stress biomarkers in the liver cell. The same thing was done. Also, very interesting point here, the dose which was active from the white flower extract was almost half of the dose from the standard silimarine which is used in the market. We did the same experiment on the oxidative stress biomarkers like MDA or glutathione or superoxide dismutase, and we get the same result. The white flower extract alone was safe to the cells, produced again dose-dependent decrease in the oxidative stress biomarkers. And finally, the histopathological data confirmed our results that the white flower, which is the rare form of this plant, is more active than the commonly used or the commonly known silimarine all over the world. Here, the normal hepatocytes. Here, after using paracetamol in high dose, produces necrosis in the liver cells and infiltration of macrophage. White flower extract actually in a low dose produced a very good results, similar to the purple flower extract. Finally, I'd like to acknowledge many people have worked with me in this field. Professor Tony Guido from United States and his UIC and his magnificent group, my colleagues in the public university in Bani Suef. I'm in sabbatical leave to another Heliopolis University, another university in Cairo. The part of this work was funded by the main funding body of scientific research in Egypt, Science and Technology Development Fund. Final word, if I have 2 minutes, I would like to, oops, doesn't come, okay. Invite all of you, your students, to attend the next summer school, which will be organized by my country in collaboration with University of Nairobi this summer 2022 in one of the very magnificent places owned by my university, actually in the desert, showing some kind of sustainable development or biodynamic agriculture of medicinal plant by reclamation of the desert and producing a very successful example of sustainable development using this kind of tools of biodynamic agriculture, and also learning from the very natural ways of treatment in here it's written in Arabic, الواحات البحرية or بحرية oasis, very far from Cairo, from most of the major cities, like 5 hours, 4 hours by car. So I'd like to invite all of you or your students if you would like to join us. Thank you very much.
I'm interested to know that the silicon material plant is very abundant in Pakistan, but the extract is coming from China and India. So it's due to the collection method. These are not collected due to bond with the plant. So is there any other method like machine which can collect seed from the plant in your country?
Well, I know what you mean. We actually had the same problem in Egypt in the beginning.
Okay.
Yeah, maybe in the 80s. I can show you here. We began the work actually to try to answer your question here. To try to find an economical and sustainable source to obtain the raw material. The real problem here was to find a method of extraction which can give silimarine or flavonoid glands in a yield almost 65 to 80%. So this was the real problem challenging most of the pharmaceutical companies. But collecting the seeds themselves from the plant by very simple mechanical machines can be available. But their problem was.
So we don't have machines to have such machines in your country.
Definitely we have it. It's like it's using a very simple principle which was used by the ancient Egypt thousands of years ago, winnowing, if you know, to separate the heavy seeds from other lighter parts of the plants. But the problem, technical problem for pharmaceutical company in Egypt, were to find or to bring the extract, or come out with an extract containing the required amount of silimarine inside this extract, which is almost 65 to 80%. We'll just answer the question.
Thank you. Wonderful lectures. Our third lecture is going to be online. Our next speaker is Dr. Dimitri Neruk. Dr. Dimitri Neruk is a chartered chemist as well as a fellow of Royal Society of Chemistry, and he is currently working at Department of Mathematics, Aston University, Birmingham, UK. He has published over 70 publications, including in Nature, Communication, Journal of Physical Chemistry, etc., and recipient of major international project grants, which each worth over 2 million. So it's a pleasure to listen to you, Dr. Dimitri Nero, and he will be talking to us on high performance molecular dynamics modeling of biomolecules at multiple space and time scales. Over to you, Dr. Dimitri.
Hello, hello. Good day. Can you hear me? Can you see me all right?
Yes, we can hear you, Dr. Dimitri.
Okay, thank you very much. Okay, thank you very much for inviting me for the talk. I'm really sorry I could not come in person. That's beyond me because of this situation with the COVID and all these problems. Anyway, so I'm from Aston University, which is a university in Birmingham, UK. This is our campus. We are in the city center and we are good university. We are located in Birmingham, which is in the very middle of the United Kingdom. So you're all very welcome to come and visit us. Few words about my colleagues and funding. I have many active collaborations at the moment. So these are people who contributed to this talk directly. And you can see the geography is quite extensive. These are the funding bodies, and that is my group a couple of years ago. So the subject of my talk is the modeling, computer modeling of biological molecules. I'm sorry, it's a little bit off the topics for the conference, but I believe it will be interesting for you. I will try to explain in simple words, starting from the basics. And you have definitely seen a lot of pictures like this. These are proteins, and in particular, this one is albumin, where we investigated this interaction with the fullerenes. These are different sites of interaction of fullerenes, which have some therapeutic effect. But the computer models of proteins obviously consists of these chains, and the main difficulty is that in order for the model to be realistic, it has to be salvated. It should be in water, and to be even more realistic, there should be a lot of water. And the problem is that for computer modeling, most of the resources are used for modeling water molecules, which are quite often not the main subject of the investigation, but you cannot do anything. You need to include all these water models in your simulation. And then when it comes to the theory of molecular simulations, the classical molecular simulations based on the very well-known formula. Only, of course, we need to solve these Newton equations for a very large number of atoms. So these are equations of motion where R is the position of an atom, F is the force, M is the mass, but we are talking usually about 10 to the 7th number of such equations to solve. And the essence of the model is encoded in the so-called force field, which is a potential energy of interaction between atoms. There are a number of assumptions and simplifications, and one of the main fundamental assumptions that the potential energy of the whole system is assumed to be equal to the sum of pairwise interactions. So we are only talking about 2 atoms at the time, and the total energy is the sum of those pairwise interactions. And the further simplification is that those pairwise interactions are also central forced. So it's not important the direction of the distance, only the distance, the absolute value of the distance. And of course, one of the most used interatomic potential, the so-called Lennard-Jones potential, which has this simple analytical expression, and it mimics the true potential energy of interaction of two argon atoms. It has two parameters, sigma and epsilon, which define pretty much the chemical nature of the atoms. It defines the equilibrium distance between the atoms and the strength of the interaction. But these are theoretical equations of motion. When we start talking about the computer modeling, we resort to the numerical methods for solving those Newton equations, and they are translated into simple difference equations like that, which can be coded in basically few lines of code. Of course, the results depend very much on the initial positions of the atoms, and that's the main problem when we start computer simulations. Luckily, recently, the developments of crystallographic techniques and especially cryo-electronic microscopy techniques are so great that we can now measure the positions of atoms, of every single atom, for very large biological systems, like, for example, complete viruses or complete cellular organelles. And this is a great help, and the results of simulations depend very much on the quality of the initial conditions for starting the simulations. So those are the basics. I'm sorry if I was too simple. I hope that will help you in understanding what we do, actually. A couple of applications. So one of the most striking application from my point of view is the simulation of protein folding. Despite all these simplifications and assumptions and the classical nature of interactions between the atoms, such simulations are capable of folding small proteins in solution. In other words, you put in your simulation system unfolded protein chain, and then after a few million steps, you got the native state of the protein. This is amazing, and this example is a bit out of date. That's 2014, and nowadays much more proteins are folded. Another application which we developed quite a lot is the simulation of complete viruses. So this is an example of experimentally measured structure of a small virus called PCV2. And this is the first example that's been done in 2011 by Reza Hayat, with whom we collaborate actively up to this day. And he has measured the atomistic structure of the capsid of the virus, that is the protein shell of the virus. And we use these measured structures to build the molecular dynamics computer model and to simulate it for long enough time. So this is the protein shell, the capsid, and surrounded by the water solution, including ions. I'll talk about this a little bit more in a couple of slides. Another example, which we do in our lab, in our group, is the simulation of catalase. This is the main target in tuberculosis bacteria, and the antibiotic which is used in curing tuberculosis is isoniazid. That's a small molecule like this, and it's known to target catalase. That's one of the functionally critical protein in the bacteria. So what we have done is we simulated the process of dissociation of this antibiotic isoniazid from the catalase. So these are the active sites, and after several seconds, the antibiotic diffuses out of the catalase. What's interesting is that we have obtained a very good match with the experiment. The technique is a little bit more involved. It involves several simulations, actually many simulations, with different value of the random force, artificial random force applied. So if you plot these dissociation times against the random force, you can extrapolate to the zero random force, which corresponds to the experiment. And we have found exact match, pretty much numerically exact match. We obtained the value of 36 seconds, where the experimental value is 50 seconds. And I mind you that in simulations, we are talking about picoseconds and nanoseconds, which is 10 orders of magnitude smaller than the experimental value. And despite that, we have achieved a very good match. Another application is the development of the so-called nanotubes, which is the composite material made of small proteins like these, small peptides rather, and the graphene oxide. So we collaborate with a group in Nottingham who specialized in making these materials, and the point is that they can make very small tubes, microtubes, I would say. The big advantage is that those microtubes are biocompatible. That's in contrast to many other graphene materials, which are usually toxic for biological systems. And what we have done is we have simulated the interaction between these peptides and the graphene oxide, and we have found that the interaction is happening through the formation of few hydrogen bonds, and that was actually in contrast to what the experimentalists assumed. They thought that the interaction is hydrophobic, which is not the case. And that helped us to explain, to rationalize the formation of this material, because there is a subtle balance between the strengths of the interaction and the agility of these peptides. So I will spend a few more words on simulating another virus called MS2. MS2 is a bacteriophage. It infects E. coli, and it's very well studied. So, for example, recently the atomistic structure of the whole phage, whole virus like this, has been measured, and it consists of a protein shell, the capsid, and the RNA molecule inside, which is the genome of this virus. And it has very interesting mechanism of infecting the bacteria. So what happens is that it uses the bacterial F. pilus, which is a very small tube like this, which the bacteria use for transferring the genetic information between the bacteria. So inside this tube, there are small DNA plasmids which travel through the tube to the other bacteria, and it's the main mechanism for transferring the antibacterial resistance information, which means it makes this very important mechanism to study. But what the virus does, the phage, it is attached to this pilus. So those blue spheres are the viruses. And when these pilus retracts inside the bacteria, the virus transfers its genome through the bacterial wall inside the bacteria. So people know that this happens, the biologists know that this happens, but they don't know how exactly. They know the mechanism. So what they observe is that at one moment, the phage was attached to the pilus like this. So this blue sphere is the capsid, and this schematic DNA inside is, RNA inside is the genome. And then at the next moment, the capsid remains outside, empty, and genome is being transferred inside the cell. So nobody knows how this happens, but this is important because that would allow us to construct artificial systems like that to use as a drug for killing the bacteria. What we did, we used this information and we simulated the whole virus, but the problem was that simulating the capsid was relatively easy because the atomistic structure was available for the whole capsid. Whereas for the genome, for RNA genome inside, only a limited structure was available because of the resolution of the cryo-EM image. And what was measured is the backbone of the genome, not more than that. And what we did, we have reproduced, we have fitted the chemical structure of the viral RNA into the available backbone. And after that, we fitted it inside the measured capsid, and this is the whole picture. So this blue shell is the protein capsid of the virus of MS2, and these golden double helices is the genome, the RNA. So that's for the first time when this model is built, and it's complete. So we have solvated it in water and ions. So we solvated the whole thing in physiological solution, and we did the molecular dynamic simulations for the whole thing for enough time to see that the structure is stable and it reproduces the experimental and measured structure. And then we moved on and we investigated how this virus interacts with the bacterial pilus. So that's the bacterial pilus. And we investigated how this moves. So bacterial pilus retracts inside the bacteria, and it rips off the viral RNA, the genome, from the capsid and transfers it inside the bacteria. So we know now how this happens, and we have these atomistic details of the whole process. So if you're interested, we published recently a perspective on all atom molecular dynamic simulations of whole viruses. So you're very welcome to have a look. Now, obviously, this is great, but there are quite a few problems. As I said, most of the resources of the computers are spent on modeling water, essentially. And this is a bit wasteful because we don't need the information on water. We're only interested in information in the information of biological molecules, proteins, DNA, RNA, and so on. And it happens that we cannot increase the size of this model indefinitely, because there are estimations that if you want to simulate using molecular dynamics, for example, a cell, right, in the entirety, the cell, it's impossible even theoretically, because even you assume that you have infinitely many processes with speed of light communication between them, which is obviously the theoretical limit, then even then you would need hundreds and hundreds of years of simulation to do the whole cell. Therefore, something more intelligent is needed. So the models should be smarter and more intelligent. Yeah, okay, so this is the simple graph which represent the current state of affairs. So there are, that's the K computer, the Japanese machine, which actually been retired last year, but it roughly tells you what are the limits of current time and space scales for simulations. So to make the model more intelligent, more knowledgeable, what can be done? All these water molecules, which we don't actually need, can be replaced with something more economical, and that more economical is the hydrodynamics. The hydrodynamics represents water as the continuous matter, and as it is, it's much more economical to simulate. So our goal was to combine the continuous hydrodynamic representation, which is economical for simulate, with the atomistic representation of those parts of the system which we are interested in. Yeah? And there is a fundamental problem with that, because these are two fundamentally different representation of one and the same matter. Because, as I said, the hydrodynamic water is the continuous fields, the fields of density, velocity, and temperature, while atomistic representation obviously is the collection of coordinates and momenta of all the atoms in the system. So what we have done, I'll skip this, I don't have time for that. What we have done, we have developed a theoretical framework for correct combining these two representations. So what we have in our system is a part of the system which is described using atomistic molecular dynamics, where we have all the atoms explicitly represented as in the usual molecular dynamics. And then we have another part of the system which is continuous hydrodynamic representations. So there are no atoms there, there are only fields which represent our solution. And we have a phase, the region in space where these two representations are joined together. And the subtle point here is that this joining has to be physically meaningful in the sense that the fundamental conservation laws are satisfied. That are the conservation of matter, the conservation of momentum, and the conservation of energy, right? So that's not trivial to do. We have done that. We derived these equations. I will not go into the details. I'll just mention that we have developed the theoretical framework, and we implemented this framework in the standard molecular dynamics packages like GROMACS. So this is an example of such hybrid multi-scale model of a virus. So these blobs here are the atomistic representation of the viral proteins. So if you look closely, you can see that every atom is represented explicitly. Then around these proteins, we have a shell, a layer of atomistic water, because water needs to be represented atomistically when it's next to the proteins, to the biomolecules, because it interacts with the biomolecules as molecules. We cannot replace them with the continuous, because that will introduce large artifacts. So we have this atomistic layer of water, and then we gradually change this atomistic water into the continuous environment. So these blue balls are the hydrodynamic representation of water, of the solution. And that makes the complete hybrid system, which is physically sound. So as I said, we have implemented this in GROMACS, and we run the simulations, and it shows that it works well. Okay, 1 minute. We are now working on some other presentation, which is more economical and more straightforward, but I guess I will not get into these details, and I'm ready to answer the questions. Yeah, thank you.
One question if anybody has. Yes, 1 minute.
Hi, Namaste. Thank you very much for your presentation. It's very nice. Good experience. Namaste, if an identical system and if they run with hydrodynamics and molecular dynamics, how you will compare the results of both systems?
Oh, sorry, I can't hear you properly. So can you repeat, please?
Yes. If we have identical system and apply molecular dynamics and hydrodynamics, how you compare the two dynamics?
I'm sorry, which system did you say?
If we have one system.
Yes.
And we apply hydrodynamics, and in the second, we apply the molecular dynamics.
Yes. Okay. So that's the whole point. So we combine the representation as, so we have two phases of the same substance, same matter, right? Water, for example, yeah. And we say that our water, our physical water, is represented by the mixture of two phases, the molecular dynamics and hydrodynamics. And there is the parameter S, which tells what is the fraction of each phase. So if S equals to 1, then we have purely hydrodynamics. If S equals to 0, we have purely molecular dynamics. And that is only the theoretical model. Obviously, physically, it's one and the same water, right? But theoretically, that's represented by two different equations of motion, and they are correctly combined. Thank you.
Thank you, Dr. Dimitri. Thank you very much for your valuable time, and we hope to see you next time at the TJ. Thank you.
Thank you.
So now we will end up this session, and we'll have a tea break of 20 minutes, and we should be exactly come back here by 3:55. Thank you.